senior 1 in a new lesson. Okay, we had before first lesson about carbohydrates, second one about the proteins, and third one about lipids. Okay, and you can output them. Okay, and the last one in macromolecules about nucleic acids. Nucleic acids, as we said before in first lesson, have two types, RNA and DNA. So in our daily lesson, okay, we will explain the difference between the structure of DNA and RNA and the function of both of them. First one, DNA and RNA, both nucleic acids, macromolecules or polymer. Macromolecules or polymer. Any polymer consists of a monomer. Okay, this is not a resonance point. This monomer here is its name nucleotide. So nucleotide is the monomer of a polymer. This polymer is a nucleic acid. First of all, I want to know the structure of this nucleotide. And same in RNA or DNA or not, we will see later. Nucleotide structure. Nucleotide consists of, first, this shape is sugar. Okay, so the first part in a nucleotide, sugar. Okay, sugar consists of 1, 2, 3, 4, and 5 carbon. So the type of sugar here is a pentose sugar. How can I count the number of carbon? With the clockwise. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So the first structure here is a pentose sugar. The type of pentose sugar here is a ribose sugar. Number 2, phosphate group. Phosphate group, its location is conjugated with carbon number 5, PO4. The type of bond here is a covalent bond. So the covalent bond binds between carbon number 5 and phosphate group. Number 3, carbon number 1 from this side binds with nitrogenous bases or nitrogenous base. Okay, nitrogenous base with the same bond, covalent bond. We have here nitrogenous bases. We have 5 types of nitrogenous bases, okay? Look carefully at me. A, C, G, T, and last one, U. A, adenine. C, cytosine. G, guanine. T, thymine. U, uracil. Okay, so we have here 5 nitrogenous bases, A, C, G, T, and U. All of them? No. One of them, okay, bind with the carbon number 1 and form one nucleotide. Okay, this is the structure of any nucleotide. But same nucleotide in RNA and DNA? Of course no. Okay, what's the difference between the structure of nucleotide in DNA and RNA? Please look carefully. Nucleotide structure, this time, but of DNA. Number 1, sugar, as I said before. The type of sugar is ribose, but look here, ribose not completed in structure. It's free of one atom, free of one atom. You said before, sugar carbon, hydrogen, and oxygen. And the relation here which control me, N to N and N. Carbon is 5, hydrogen is 10, oxygen is supposed to be 5 also. No, oxygen here 4, not 5, because of elevation 1 atom of oxygen. So the type of sugar here, not ribose, but deoxyribose, meaning of D without oxygen, ribose sugar. C5H10O4, this is the structure of the deoxyribose. Number 2 is same and constant in both nucleotides. What? Phosphate group is same in both types of the nucleotides. The last one, number 3. The last one, nitrogenous base. Nitrogenous bases, DNA contain 4 of 5. 4 of 5. Look here, adenine with me, cytosine with me, guanine with me, and thymine with me. No uracil, okay? Adenine, cytosine, guanine, and thymine, okay? So the type of bases here, adenine, guanine, cytosine, and thymine. Okay. What is the shape of the DNA? DNA like in this shape. Okay? Like the ladder. Okay? Like ladder. Ladder consists of 2 sides, 2 strands, and between them drags. Between them steps. So the strands here, 2 strands, and between them steps. Strands of DNA is sugar plus phosphate. Steps, okay, nitrogenous bases. Nitrogenous bases. So we have here 2 strands. Can I connect between first and second strand? Yes, between those nitrogenous bases. If here nitrogenous base here is adenine, so must the opposite one be thymine. If the first one is cytosine, have or must the second one be guanine. So we have here binded must be formed. Okay, adenine must be bind with thymine, and cytosine must be bind with the guanine. Okay, look here, adenine with the thymine, guanine with the cytosine, adenine with thymine with the dicovalent bond, guanine with cytosine with the triple covalent bond. This is the bonds between the 2 sides, 2 strands of the DNA. Now we are going to explain the structure also of nucleotide, but for RNA. RNA consists also from sugar. The type of sugar here, not deoxyribose, but complete ribose. Ribose sugar. So the structure of ribose here, C5H10 and not 4, but 5, complete ribose. Number 2, phosphate, sugar. Number 3, nitrogenous bases. The type of bases that present inside the nucleotide of RNA, adenine with me, cytosine with me, guanine with me, thymine no, instead of it, uracil. So we have here 3 old and 1 new. 1 adenine, guanine, cytosine, and uracil. Okay, this is the structure of the RNA nucleotide. Okay? the difference between DNA and RNA in structure? The nucleotide of both of them, okay, this table can DNA, RNA. Here, nucleotide. First one, sugar in DNA is deoxyribose. Here is ribose. The structure here C5H10O4. Here C5H10O5. Number 2, phosphate. Present in both. No different. Number 3, bases, nitrogenous bases, okay? Nitrogenous bases, okay, here A, adenine, guanine, cytosine, and thymine. Here, adenine, guanine, cytosine, okay, and uracil. From this structure, what can I explain the main structure of nucleic acids? The elementary stages of the nucleic acid consist of, like any organic substance, carbon, hydrogen, and oxygen. Here, phosphate, P, phosphate group, PO4. So we have here a new element, P. Nitrogenous bases, from its name, contain nitrogen element. So the first one is nitrogen. And please here, focus. This structure you said before. Yeah, you saw before in lipids. CHOPN. Okay, you deal with it before in lipids. Phospholipids. Phospholipids. And please focus, this is very important point, in protein like casein. In protein. So, in structure is coming between 3 parts inside your curriculum. Okay, nucleic acids, protein, and lipids. Okay. Function of DNA. DNA consists of genes. Okay, each group of nucleotides consists of 4 genes. So, gene is group of nucleotides. Each gene is responsible for what? Responsible for traits. Meaning of traits, inherited traits that we are get from parents. Traits to be appeared in our features have to resemble by protein. So, trait is a protein. To appear the trait, you have to do protein. Protein to be formed by RNA interference. RNA gets from DNA. So RNA gets from DNA. Meaning of get from equal transcribed or copied. So RNA is a copy from one strand of DNA. If RNA, this is very important diagram, transcribed from DNA. After transcription, RNA, first step. Number 2, RNA translate, yes, translation. Translate what? DNA. After translation, number 3, production of protein. Production of protein. After production of protein, trait is appeared. So this is the sequence, the interaction between RNA and DNA. RNA transcribed from DNA. DNA previously consisted of genes. These genes is trait, okay? But after translation to protein, how can I translate translate this gene to protein by the aid of the RNA. Okay, so the difference between DNA and RNA: DNA double strand, RNA monostrand. DNA consists of phosphate, okay, similarities, and RNA also consists of phosphate. The type of nitrogenous bases is different between those nucleic acids. DNA contain 4, like adenine, guanine, cytosine, and thymine, while RNA consists of also 4, but different in one: adenine, guanine, and cytosine, and uracil. Okay, DNA and RNA complete their mission of each other. Okay, DNA is present inside the nucleus without getting out, and RNA transcribed from DNA and get out from the nucleus and present in cytoplasm. Okay, both have same mission. DNA and RNA macromolecules, they both of them consist of nucleotides, so symmetrical in shape, symmetrical in structure, while different in structure of nucleotides. Okay, thank you, everyone, and see you again, inshallah, in the next video. Thank you.